Yes, definitely. So autophagy is the process by which cells can digest or break down waste material, like proteins that accumulate. They are broken down into individual amino acids through autophagy, but also large cell compartments like mitochondria are broken down through autophagy. It's called mitoautophagy. You have different forms of autophagy, macroautophagy, which is good to break down large cell compartments or organelles like mitochondria. You have then a microautophagy, and you have chaperone-mediated autophagy, which actually can tag specific proteins or materials for breakdown. So autophagy is very important for longevity because you can compare it to the waste incinerators of the cell. So our cells produce a lot of garbage every day. You need to get rid of the garbage, and they put it into the waste incinerators, like the lysosomes, and that's through process like through autophagy, and also through other very related processes like ubiquitin proteasome complex, which is mainly specialized in breaking down protein. And then you have macroautophagy that breaks down more the larger stuff. And so anyway, super important for longevity, because the more autophagy, the more you break down waste that otherwise would accumulate and contributes to aging. Like Alzheimer's disease, you have accumulation of proteins inside the brain cells and outside the brain cells. And that is probably one of the driving factors behind Alzheimer's disease. So breaking down this protein accumulation and other waste materials is very important. And we've seen a lot of studies, if you upregulate autophagy, you extend lifespan and improve health span.
These signaling proteins, like the DAF2 one that Cynthia discovered, and the sirtuins that we work on, we didn't know what they were doing at first. It was like, this is crazy. It's got nothing to do with DNA repair and free radicals. But then when we tracked what they were actually doing, it turns out they're speaking to the rest of the cell to keep the cell healthy and alive. So in the case of that worm, those worms weren't just longer lived. The program to survive was activated. Those worms are stress resistant. You can hit them with heat. You can hit them with cold. You can starve them. Or just put them in happy conditions, they live longer.
What we didn't expect was the extraordinary propensity for survival. We could leave the cells out and put them in buffer. They wouldn't die under extraordinary circumstances that we couldn't explain. So why would a cell that just couldn't commit suicide survive in buffer with no nutrients at all? We puzzled over this, like, how can this be? And then we discovered that what these cells had done was turned on autophagy, and were using that for survival, and that this was a mechanism by which yeast survived starvation. So we engineered mice where we can take an adult mouse and give the mice a chemical so that an essential autophagy gene will be deleted throughout the entire animal. So one day they're an adult mouse with autophagy, and then a few days later they're an adult mouse with no autophagy. And these mice were very extraordinary. They lived for 2 to 3 months, and then they died predominantly of neurodegeneration. Autophagy is very important in the brain over the long term. One other way of looking at it is what tissues are more autophagy dependent than others. Brain would be really important, and there are a few others. And what we noticed when we looked at the mouse that lacked autophagy was that there were tissues like the brain that were very sensitive, and there were other tissues like the lungs that didn't have any phenotype.
There are all kinds of substances found in nature and in food that can induce or upregulate autophagy. For example, fisetin is a substance found in strawberries. It's also often called a senolytic, which actually also very good anti-inflammatory, by the way. But fisetin also can upregulate autophagy through inhibition of mTOR, because mTOR, if you activate it, you downregulate autophagy. So if you eat a lot of protein and then you have a lot of amino acids in the bloodstream, amino acids activate mTOR, downregulate autophagy, and so you have more cellular waste material accumulating inside and outside of cells. So fisetin is an mTOR inhibitor and can that way induce autophagy. Pterostilbene also could induce autophagy. AKG, alpha ketoglutarate, we just discussed, can, according to some studies, also induce autophagy. Microdose lithium. So we've seen a lot of studies that actually low doses of lithium can extend lifespan. Actually, in cities or areas where there is a lot of lithium in the drinking water, we have seen association studies showing that people tend to live longer and have less neurodegenerative diseases or less suicide, by the way, because even low dose lithium can serve as a mood stabilizer somewhat. But it also induces autophagy. But actually, besides induction of autophagy, you also need other, let's say, mechanisms to slow down this accumulation of proteins and other mass. Like chaperones, we discussed like glycine earlier. It's a chaperone because it protects new proteins against accumulation. And you have also microdose lithium, for example, can also upregulate autophagy, like we said, but can also activate all kinds of genes through epigenetic mechanisms that can improve proteostasis. So that's the homeostasis of the proteins and preventing them from accumulating too much. And then you have also substances that can also reduce the protein accumulation through activating a specific response. It's called the unfolded protein response, like acetylglucosamine, which is a component of hyaluronic acid, by the way. If you upregulate through, for example, acetylglucosamine, the unfolded protein response, you also induce autophagy and you help the cells to better maintain proteostasis, or let's say the protein metabolism.
Spermidine is found throughout life. It is found in sperm, but it's also found in plants. It's found in our bodies, and it's called a polyamine. It's a really small molecule that is actually very protective against things like oxidative stress. And one of the things that it's been found to do mostly is to induce autophagy, the ability of the body to recycle old proteins and make proteins back into amino acids to build new proteins, which is very important for longevity. And Frank Medeo, who certainly you know and I know, he's a scientist from Austria, has done about 15 years of really high quality research looking at starting with yeast cells where there's spermidine, but also in worms and flies and mice, and increasingly some studies in humans that we're aware of that is not yet published, but we're going to share that with you. Mm-hmm. This molecule extends the lifespan of all those species. And that's really amazing. There are very few molecules that I'm aware of that does that. So I've recently started taking spermidine as well, because we want to induce autophagy, and that would help induce what fasting would normally do, or it would help accentuate the benefits of fasting.
And autophagy is getting rid of damaged cells, and we have to get rid of damaged cells to heal and recover. As we age, we get more and more and more and more damaged cells, or what we call senescent cells. Senescent cells are cells that are designed, they're basically, so cells age, they start getting filled more and more and more with trash and damage. And at some point, they're designed to kill themselves, recycle the good parts, and get rid of the bad parts and go away. If they don't, they stick around. I like to call them zombie cells because they actually spew out proteins and they actually damage the cells around them. So senescent cells will create more senescent cells, will create more senescent cells. So part of longevity is getting rid of the damage. And spermidine is an autophagy-inducing agent. Fasting is as well. So spermidine is a fasting mimetic in that it actually gets rid of these damaged cells. It particularly seems to work well with mitochondria. So you get what's called mitophagy, you get rid of damaged mitochondria. So spermidine, if we talk about that whole mitochondrial biogenesis, right? I want to make new mitochondria, but I got to get rid of the old guys. The sick old guys are not doing any good. They're taking up space. They're utilizing resources. I got to get rid of them. So spermidine is a really nice option for that. It has its best benefits for autophagy by using a high dose. So I like to do a high dose periodically, like let's say 4 times a year. You do a high dose, like 6 milligrams for 30 days. And then you can use a maintenance dose just on a regular ongoing basis, which has more of a just keeping things level, but not a big sort of die-off. So it's kind of like cleaning house. Every now and then you got to clean out everything, right? Just clean out, spring clean, and then you got to keep everything okay. But even if you're keeping everything okay, eventually you got to go back and spring clean. So that's sort of the concept of using spermidine at a high dose periodically and a lower maintenance dose.
Yeah. Absolutely floored, because it does things that none of the other agents can do. It just fills gaps that we needed to fill. It's incredibly good for preserving cellular function, which is what sperm needs because they're in physiologically very stressful conditions. That's just the way that it is. But in terms of what it does for us, and I won't go through everything, but I'll go through the highlights. So in the epigenetics category, it basically inhibits something called P300, then activates autophagy. So it is one of the strongest activators of autophagy that we have. Something unusual because we have things that kind of do it a little bit, but not really as potently. So this is huge in our arsenal of autophagy. I sort of decided that this was sort of like bubble wrapping your DNA, just protects your DNA. And that is something we have not seen in any other molecular agents. Here's a big one I'm missing. It actually helps with the mitochondrial transition pores. So mitochondrial transition pores are pores in the outside of the mitochondria that open more frequently with age. And they leak mitochondrial contents into the cell. It destroys the electrochemical gradient, and you get all the sort of cytotoxic stuff going out into your cell. And it's but spermidine actually helps to control that. Where was I? Ah, yes, hair. It's so cool. It stimulates stem cells in the hair follicles. So it absolutely does help with hair. So I'm convinced by the time you you take your spermidine, you take your Clonia cava, you're going to have the fullest head of hair ever.
So that was the situation there. So we evaluated only the sirtuins and the PARPs enzymes, and we saw that there is definitely a link between those being hyperactivated because of all of this DNA damage that is going on in neurodegeneration, and then the NAD consumption, right? Mm-hmm. But interestingly, if the sirtuins and the PARPs are activated nonetheless, and we feed those cells with NAD boosters such as NR and NMN, we will still see a beneficial effect. So this is interesting because the fact that your autophagy in your brain will be going down with age is inevitable, right? So what do we do when it comes to our daily brain maintenance, and why should we supplement with NAD boosters? Well, this is a very interesting case that even when autophagy is going down, it could be very beneficial to supplement with different NAD boosters. And there are studies that are showing that at least for NMN, I haven't looked into NR, but at least for NMN, it looks like there are at least 2 studies that are demonstrating that NMN can be blood-brain barrier permeable in mice. And basically what happens is that the Alzheimer's or dementia phenotype in, for example, Alzheimer's mice is being rescued by NMN supplementation. Now,
the next one, let's talk about, which a lot of people call intermittent fasting. We'll just call it fasting. This is a period if you go longer than a day. Some people do 3 days. Some people go for a week. I wouldn't go longer than that because then you'll start chewing up your muscle, which you don't want to do. But these long extended periods are doing a real deep cleanse on the body and turning on that autophagy, that process of recycling proteins very deeply, especially once you get beyond the 3-day mark when your metabolism switches into what's called chaperone-mediated autophagy, the deep cleanse. So that's fasting. And then there's time-restricted feeding, which is what I do because I'm not very good at going beyond 24 hours.
Well, I think most people would really, really struggle to get there.
Yeah, time-restricted feeding, which is what I do, which is try not to eat till dinner. That's hard enough. My hat's off to people like Peter Attia, the doctor and podcaster who many of our audience will have heard of. He can go for a week, and he's got a lot of willpower. If you've ever met the guy, he's different than me. I'm more of a hedonist, but
Is he grumpier at the end of the week, do you think, than he is at the beginning of the week, though? I mean, like after all of that time?
He's always serious. That's what I can tell you, but brilliant. But let's get back to this time-restricted feeding. You want to have at least 16 hours of not eating or not eating very much. And then you can have 8 hours. So typically that means having a late lunch if you skip breakfast. Or if you prefer to skip dinner, skip that.
Because what we got to remember here is that there's a period of sleep in which you are not eating. And
that's the easy part.
What you're really doing is tacking on hours to the front of that and tacking on hours to the back of that.
That's right. So when people say I'm skipping lunch, that's not as helpful. And so what I've done in my life, let's just use me as an example of an average human being, because I'm pretty lazy and I'm not that driven. I prefer to take a pill, which is not the right thing. What I started out in my life was not to eat breakfast. And I've pretty much been doing that since I was a teenager. And that for me works because I'm not really hungry in the morning. I have a cup of tea or a cup of coffee, and I still do that. A little bit of yogurt to mix my supplements with to dissolve them, but that's
and you're not doing the yogurt. And I've actually seen you do this. That like the amount of yogurt that you put into your little bowl is really minimal. It's basically just to mix up
a couple of spoons.
Yeah,
that's it, and that's burnt off in 15 minutes. That's not going to be a big deal for the rest of the day. And I try not to eat at all until dinner, and then I have a really enjoyable, largish dinner. But it's certain type of food. We'll talk about the types of food later. But that gives my body this long window, more than 20 hours of not having glucose circulating from the external world. Now, what happens when you do that, and it takes a few weeks for your body to adapt, is that your liver will learn how to compensate for a lack of food. It's called gluconeogenesis, the generation of glucose from your liver, and it actually overcomes the feeling of hunger. When you first skip breakfast and lunch, you're going to be hungry. You're going to be nervous. You're going to say, I can't do this. Sinclair's an idiot.
Well, this is why people fail at this, is because they run into that and they think this is how life is going to be for the rest of the time that I'm on this diet. I can't do this diet.
Right. But do it for at least 2 weeks, because after the 2 week, especially by the 3 week mark, your liver has now learned that you're not going to have breakfast or lunch, and it will start making glucose at a steady level. That's really important because it's known that if you have these spikes of glucose, it leads to hunger when it crashes after a big meal, but also that when you get hungry, you eat. So you're in this wave of hunger, eating, wave of hunger and eating.
Do you think
You have no breakfast, no lunch?
No.
Because
Well, because it's clear based on thousands of years of human existence that eating less is, if it's not starvation or malnutrition, it's good for you.
because why? What does
Well,
it do?
so we didn't know why it was good for you. We thought that it was slowing down metabolism, but it's not the case. What's happening is it's turning on, when I'm not eating, or if you choose not to eat for a meal or two, you're turning on the body's defenses against aging.
In our labs, we always have caloric restricted animal. Okay,
Yeah.
you take brothers, two groups, one eat whatever they want, the other gets 60% of that. And they
Okay.
live 40% longer and healthier. And people, and we know it since the 1950s, but people assumed that means that you give less for breakfast, lunch, and dinner.
you just eat less throughout the day
Right.
as opposed to, but that's not what it is though, right?
No. If we actually are giving them all the food throughout the day, they're thinner, but they don't live longer. So you need the fasting to upregulate the mechanism for aging.
Well, the trick to longevity is a term called hormesis, which is a little bit of stress on your body, biological stress. So running, bit of hunger, cold, heat, puts your body in a state of perceived adversity. So you don't have to hurt yourself, but you put your body in a kind of a scared state. Oh my goodness, I could die if this continues. And then they turn on these defenses against aging. So we have these genes that get turned on. Some of them that we work on are called sirtuins. And so by being hungry, we've shown that you turn these on and then your body defends itself. If you sit around and you eat constantly, they don't work very hard.
Really?
And your clock ticks faster.
In terms of exercise, that's very well studied, that exercise induces autophagy very potently. And you actually need autophagy because exercise damages the muscle. And autophagy is one of the processes that helps mitigate the damage that occurs during exercise.
What about hypoxia?
Oh, potently. Hypoxia potently induces autophagy.
Hypoxia, the low oxygen when you go for a run, what it's doing is turning on this HIF-1 alpha protein that I talked about earlier. And that helps promote health in the body. Hyperbaric chamber treatment does the very similar thing. You get a production of these free radicals, and that stimulates also a mitohormesis response that gives you very similar benefits to low oxygen. There's one theory that I have is that when you come down from the high levels of oxygen, it simulates hypoxia, like you're running as you come down from high levels to low levels. It's the differential, and that fits with the new findings, which is going up once and coming down once is not as good as going up and down within a treatment.
I specifically sort of designed my workout routine based off of what I think are going to give me the biggest brain benefits. And what I've sort of come to the conclusion of is that intensity does make a difference with respect to the neurobiological effects. Exercise intensity. The biggest and most compelling and most important data point that I sort of learned from wearing my continuous glucose monitor was the effect that my sleep interruption had on both my fasting blood glucose. I could get like what can be considered like pre-diabetic. Like I was like blown away. I was like, this is insane. And the effect lasted at about, I would say like about 48 hours or so. When I did work out, and at the time I was doing a lot of high intensity interval training. It was like an hour-long spin class I used to go to, where they do all this interval training. That it almost completely blunted that effect, where even though I was dog tired, last thing I want to do was go to my damn spin class. I was just like, this is like, I can't, it's going to be bad for me if I go. That's how I felt. Like it's going to be bad for me. But it was completely the opposite, where crazy glucose, dysregulation, and it was almost completely blunted. And it was so profound. And really, that was the thing that for me was like, I have to work out no matter what.